These are the recorded lectures for chapter 29. So this is the second recording, and we're starting with slide number 15 in the chapter 29 PowerPoint slides. What we went over in the previous recording is that there are three events that lead to genetic variation, and that is independent assortment, crossing over, and random fertilization. I know it says crossover here. When I teach this in biology, I've always said crossing over. So crossing over, crossover are going to be the same thing. Okay, but let's talk about independent assortment. So, the key down here at the bottom says that the purple chromosomes are from the father and the green are the maternal or mother's chromosomes. So what you're looking at, this original cell that has 6 chromosomes or 3 pairs of homologous chromosomes, that cell is the sex cell that's going to undergo meiosis and become either egg or sperm. Okay, so what you're looking at is a stage in the cycle of meiosis called metaphase 1. And that is when the homologous pairs of chromosomes line up in the middle of the cell. And then what they're eventually going to do is separate. Excuse me. At the end of meiosis, you have 4 cells that have half the number of chromosomes, and they're single-stranded chromosomes rather than the ones that look like the letter X that are double-stranded. Okay, so you have 6 chromosomes, which is the diploid number in this parent cell, and then you have 3 chromosomes in each of the daughter cells that form after meiosis. So what is independent assortment? It is the order that the chromosome pairs line up in during metaphase 1, or it's how they pair up. Whether they pair up with the father's chromosomes on the left and the mother's on the right, or like you see a mixture, or you can even have all of the mother's chromosomes on the left and the father's on the right, but you have all these different possible combinations because each one of these chromosomes can line up independently of the other pairs. So all of the father's chromosomes can be on the left, or you can have like the first pair has the father's on the left, the mother's on the right. The second pair has the mother's chromosome on the left, and then the third pair is like the top pair. So, and then over here you have a whole different combination, and then far right you have a different combination. So the genes that are located in these chromosomes, the different versions of those genes called alleles, there's going to be a lot more possible combinations, a lot more genetic variety with this process or this event called independent assortment. That's going to increase the genetic variety. In fact, if you go back, we calculated that there are 8.5 million combinations just due to independent assortment in the gametes. Excuse me. Sorry, I'm having allergies. But, okay, so sometimes when the genes are on the same chromosome and not different chromosomes, they're linked. But because of this next event called crossing over or crossover, those linked genes can still have the same result as independent assortment because basically crossing over looks like this. Okay, so you have a homologous pair of chromosomes here, and they've got the gene for hair color, and the mother has the big H and the father has the little h. So the mother's allele is for brown hair and the father's allele is for blonde hair. And then they have also the genes for eye color. And they're linked because those 2 genes are on the same chromosome. And the mother is giving the big E allele for brown eyes, and then the father is giving the little e allele for blue eyes. So if this were independent assortment in the gametes, we could have, remember, you're going to get one H and one E in the sperm or the egg. So we could have big H, big E, little h, little e, or we could have big H, little e, big E. Those are the possible combinations you could get if it was independent assortment. But since these are linked on the same chromosomes, these genes, the only way to get those combinations would be if, when they do pair up, if portions of those chromatids, those non-sister chromatids, were to cross over and then exchange places, like you see here. And and this is what really does happen. So you end up with big H, big E, big H, little h, big E, and little h, little e, even though those genes for hair color and eye color are linked. And by the way, this is just a side note, but it's interesting. You know how you see a lot of people who have brown hair also have brown eyes? And a lot of people who have blonde hair also have blue eyes. This is actually really true, that the hair and the eye color alleles are linked. They're on the same chromosome. So it's that's one of the reasons why you do see, certain hair colors with certain eye colors. But you also see variation. You see people with dark hair that have blue eyes, and you see people who have blonde hair and brown eyes. So that is due to the crossing over. Okay, so we've talked about two things that can give us genetic variety. That is independent assortment and then crossing over. And now they said crossing over up here at the top. So the third thing is random fertilization. Random fertilization means that, okay, females don't just produce one egg and males don't just produce one sperm. Females actually produce around 500 eggs total, and one or two are released every month. Usually it's one during ovulation. Now, males produce, trillions of sperm, right? So they produce many, many, many sperm. So each of the eggs and each of the sperm that are produced have different combinations of alleles that came from the parents of the one who produced the egg and the sperm. So because the eggs and the sperm that are produced in those individuals are different, depending on which sperm fertilizes which egg, you can have different combinations. So that's all random fertilization is. Is it random which sperm fertilizes which egg? And so we take that 8.5 million, that's the variety that we can get from independent assortment, 8.5 million different possibilities of genetic variety for egg, 8.5 million for the sperm, and you multiply that together, and that gives you about 72 trillion different possibilities in the zygote once the sperm fertilizes the egg. So that is why you look at families and you can see, sometimes you see siblings that look like each other. Then other times you see siblings that have the same mom and dad that don't look a thing alike. You wouldn't know that they were related unless somebody told you. That is the reason we get such a large variety of genetic traits in individuals, is due to independent assortment, crossing over, and then random fertilization. So they didn't even, in that 72 trillion, they didn't even include the effects of crossing over. That would make it greater than 72 trillion, and different, possibilities in the offspring, different possibilities of genetics, genetic variety in the offspring. Now we're going to move on to section 29.3, patterns of inheritance. Patterns of inheritance, that is just basically how that trait is inherited. So the simplest pattern of inheritance, I just call it simple dominance. That's how I teach it in biology. But in anatomy, this textbook is calling it dominant recessive inheritance. And what this means is that, okay, we know each individual gets 2 alleles for each trait. Well, one allele is dominant, one version of the trait is dominant over the other. So if you're heterozygous, you're going to express the dominant trait, and you'll have the gene for the recessive trait, but you won't express it. You might be able to pass it on, but we'll talk about Punnett square in a minute. I want to show you one example of a dominant recessive trait. So dominant recessive inheritance or simple dominance, for example, albinism. All right, so normal skin pigmentation is represented by the dominant allele big A. That's normal pigment. And then recessive pigmentation, albinism, it is recessive, but albinism, which is the lack of pigmentation, is represented by the recessive allele little a. All right, so if a person inherits 2 big A's, they're homozygous dominant for normal skin pigment. If they inherit a big A and a little a, they're heterozygous, but they're still going to have normal skin pigmentation. It won't be a blend. There won't be, the little a will have no effect on their skin pigmentation. That big A is dominant. So any trait that's inherited by dominant recessive inheritance is like this. You've got 2 versions of that trait, and one of the versions is dominant, one's recessive. Okay? The only way a person is going to have albinism, which is lack of skin pigment, is if they inherit 2 little a's and they're homozygous recessive. Okay? So if you take 2 parents who are heterozygous, let's see if they show you a Punnett square. They do. Take 2 parents who are heterozygous for normal skin pigment. I'm going to blow this up so you can see it better. If you were to be asked what is the chance they'll have a child with albinism, you can work a Punnett square, and that Punnett square will show you the chances of them having children with different genotypes and phenotypes. So what you do with the Punnett square is you make a box, and then you make 4 boxes inside of it. Okay? So you know what? I'm just going to do this. I know it's here, but I'm just going to do it for you. Just draw it out. So what I'm going to do is insert. Okay. All right, hopefully I can do this without making a big mess. But to do a Punnett square problem, I've already started making a mess. Okay, I'm going to try it again. Okay. So to do a Punnett square, okay, so I paused it. I wanted to, it took me a while to do this, and I know this looks horrible, but this is a sad Punnett square, but I'm having to do it with the, like the mouse. I don't have this isn't something where I can use like a stylus or anything. So I apologize, but I just want you to see how you set up the Punnett square. So in this problem, we are told that both parents are heterozygous. So you set up the Punnett square by drawing a square that has 4 squares inside of it, and you put the alleles of the parents on the outside of the squares. So, for example, if they're using the father, so the father's alleles are above the columns, and the mother's alleles are beside the rows. Both of them are heterozygous, so they each are big A, little a. Now you can combine them. So, for example, in this first box at the top left, you'd have big A, big A. And now we can look at the Punnett square that's already done. I just wanted you to see how to set it up. And there's so much on this Punnett square. You have all these numbers, and that's just showing you a different way to calculate probability. But I just want you to pay attention to the letters. So for the father's alleles, big A, little a, and the mother's alleles, big A, little a, they come together and make the following combinations. These are the combinations they can possibly make. Big A, big A. Then there's big A, little a in two of the boxes. There's a big A and a little a, and a big A and a little a. You see? So it just depends on which big A you got from the father and which one you got from the mother, you know. So, but you still end up with the same thing, big A, little a, which is heterozygous. And then there's one of the boxes where the combination is little a, little a. What this tells us, it doesn't tell us how many children that these parents are going to have, and it doesn't tell us like if they have 4 children, then 3 of the children are going to have normal skin pigment, and one of those 4 is going to be have albinism. What it does, it tells us the chance of them, the chance or the probability that they're going to have children with certain traits. So by looking at this Punnett square, it's showing us that there's a 25% chance, based on statistics, that they will have a child with albinism. Okay? They could have 10 children and all of them have albinism, like this little boy here. Or they could have 2 children and both of them have normal skin pigment. But the chance, every time they have a child, the chance that that child will have albinism is 1/4 or 25%. So this is what you do with a Punnett square. It just helps predict the chance that a couple is going to have, you know, children with certain traits. So okay. Now, this is slide number 30, so I think we're going to stop here, and we'll talk about certain disorders that are controlled by dominant traits and certain disorders that are controlled by recessive traits in the next recording.